Here red drop and Emerson's enhancement effect. So here different wavelengths of light, that is red light or the wavelength of light where the absorption of light will be there, and the photosynthetic rate will also be determined based on the light energy that is absorbed by the pigments which are present in that particular wavelength of light. So in this one, when monochromatic light means here single beam of light is processed or used here at a wavelength of red light on chlorella. So chlorella is the experimental plant which have been used here. When a monochromatic light is focused or on the chlorella, a green algae, so here the photosynthetic rate was there where the quantum yield is determined by the evolution of oxygen. And here the photosynthetic yield was constant at between 600 to 680 nanometers of wavelength, whereas it was decreasing above 680 nanometers of wavelength. So here this is a wavelength of red light where the photosynthetic yield was decreased, which is determined by the evolution of oxygen molecule. So here this decrease is called as a red drop, because here it is a wavelength of red light where the photosynthetic yield was decreasing or photochemical yield was decreasing. So this is called as Emerson's first effect, and at the same time, so he had done one more experiment also where he had provided the shorter wavelength of light of red light and longer wavelength of red light simultaneously. Both of them had given at a time. So he had observed that a sudden increase in the rate of photosynthesis or the photosynthetic yield was increased. So the photosynthetic yield was increased, which was more than that of the sum of the individual wavelengths of light which is provided to the plant. So this shows that there was an increase when both the wavelengths of light are simultaneously being operated. So this is called as a Emerson's enhancement effect. So in this process where the two photosystems were operated, so this is a way how the presence of two photosystems was discovered by Emerson in this process. And here there are two concepts have to be noted. Those are called as a quantum yield and other one is quantum requirement. So quantum requirement is the number of photons of light required to release oxygen molecules. So this is called as a quantum requirement, and the quantum yield is, so here release one molecule of oxygen, whereas in the quantum yield the number of oxygen molecules which are being released by utilizing the quantum of photons is called as a quantum yield. So here how much quanta is required for this one is a 8 quanta. So that is the number of oxygen molecules released per 8 molecules of photons, or here it shows that number of molecules will be 1 oxygen which is released by 8 photons, so it will be 0.025. So likewise, the quantum yield and the quantum requirement are being demonstrated by using the concept of number of photons and the number of oxygen molecules which have been released by absorbing that quantum of photon of light.